Hello. I would like to thank the Gairdner Foundation for this award. I'm very proud to be a recipient of this prestigious award. What I'm going to do today is to go through and describe our studies that took place in about 2 decades, over 4 decades now, on the discovery and the structure and the activity of the GLP-1 peptide. First of all, we have to ask the question, what is GLP-1? And it's shown here is the structure of the peptide hormone. It's a 30 amino acids in length. It's made in the intestine and released with meals, and it goes under the terminology of a glucoincretin hormone. And its function is to augment glucose-responsive insulin secretion and lowers blood glucose in type 2 diabetes. First of all, we need to review the physiology and anatomy of the endocrine pancreas. The pancreas is shown here in pink. It buds off of the duodenum from the intestine and consists of 2 tissues. One, the acinar tissue, which are the cells that make enzymes to digest food. And the other is the endocrine pancreas, which is shown in these little red spheres. There are about 1,000 of these little spheres in the embedded in the exocrine pancreas. And they're called the islets of Langerhans. And they consist of a collection of endocrine cells, which is shown here in a larger magnification. It consists of 3 types of cells: the beta cells, which make insulin, which lowers blood glucose; the alpha cells make the hormone glucagon, which raises blood glucose; and then there's a 3rd cell type shown here in red that makes a hormone called somatostatin, which is a negative regulator of both insulin and glucagon. Now, the regulation of the glucose homeostasis by hormones in the endocrine pancreas. Here we have a model that shows the pancreas and the alpha, beta, and delta cells in the fasting state, where there are no nutrients entering the intestine. The glucose is low, and the insulin secretion is low, and the glucagon secretion is much higher. Glucagon stimulates glucose release. Insulin increases glucose uptake. So there's a decreased glucose uptake with the lowered insulin. And so the glucagon is predominant in the fasting state. In the feeding state, we have just the opposite. And that insulin, glucose goes up with nutrients coming in, and insulin is stimulated, and glucose uptake stimulated. The glucagon is suppressed in the feeding state. But there's also, in addition to the increased glucose, this co-secretion from the intestine of glucagon like peptide 1, GLP-1. Now, what happens in diabetes, particularly type 2 diabetes, is that glucose is much elevated. It's hyperglycemia, it's called. And the GLP-1 is still secreted, but the insulin is very high, but the glucose uptake is not increased. It's still suppressed. is because of insulin resistance. Even though there's lots of insulin, it's not sufficient to stimulate glucose uptake. The glucagon, however, is very high in diabetes and increases glucose in the circulation by virtue of the production by the liver. Now I'm going to go to discuss the events that led up to the discovery of GLP-1. These are early studies that began in the mid-70s and lasted for nearly two decades. In the late 70s, in order to fulfill a goal that we set to explore the possible existence of the prohormone for glucagon. And we exploited a new technology that had just been discovered several years before called recombinant DNA cloning. And this is to determine the structures of proteins to do it via the decoding of the messenger RNA, which is converted into cDNA, as complementary DNA, by an enzyme called reverse transcriptase. And the cDNA can then be decoded using the amino acid code for nucleotide translation of DNA or messenger RNA into protein. The advantages are several in that it obviates the requirement for protein isolation. It expedites the obtaining complete sequence of a protein via the decoding of the DNA. And as a model system to do the cDNA cloning, we used this creature called the anglerfish from the Atlantic Ocean. And the reason we used the anglerfish as our first model to isolate the RNAs and make cDNAs from the pancreatic islets was because there had been a moratorium declared on recombinant DNA research in Cambridge, adjacent to Boston. And the reason for this was that there was a concern that the recombinant DNAs would get into the sewer system and be a hazard. So Boston didn't have such a moratorium, but the Mass General Hospital decided that to be safe, they would not approve of using mammalian tissue, but would approve of cold-blooded animal like the fish. And it actually turned out to be an advantage because in the fishes, the endocrine pancreas is a separate organ called the Brockman body. And it's right here, and which enriches the messenger RNA, which is used as the starting substance for the production of the cDNA. So the result was of our first studies was successful in that we found a copy of the RNA and in the cDNA, and it showed that it did indeed encode for a pro-glucagon, a large protein that contained the sequence of glucagon, and which is clearly glucagon and very close homology to mammalian glucagon, the sequence of which is known. And but in addition, there's another sequence which is similar to glucagon but different, and it's called glucagon-related peptide, GRP. And there was also this to fill out the structure, an N-terminal sequence, which made in continuous a protein called glycantin, which was known to be a major endocrine protein from the intestine. It also just started with a signal sequence involved in protein transport to get the secretion of the proglucagon into the endoplasmic reticulum. Also, these peptides were all flanked by two basic residues, lysine, arginine, which are known hallmark sites for cleavage, shown by these arrows, to release the peptides from the glucagon, the propeptide. So what we've recognized was that the anglerfish GRPs resembled gastric inhibitory peptide, which is called GIP, which is known was the glucoincretin hormone. And they were 60% similar, suggesting initially that the anglerfish GRPs are possible incretins. So it's shown here as the similar amino acids, and the identical amino acids are in circles and squares. So this brings the notion that GLP-1 might be a possible glucoincretin hormone, because and so the incretin concept that was one developed by Werner Creutzfeldt some years before, and that was the concept was that the oral glucose load is more potent than an equivalent glucose load given systemically. So that there was something that the oral glucose did, namely from the intestine, that augmented glucose-dependent insulin secretion. Now, next was to move ahead and into mammalian tissue. So we worked on the rat islet hormones, and there was some work was isolating islets from rats and extracting them, making the messenger RNA, making the cDNA, decoding the cDNA, and found the structure of the mammalian proglucagon shown here. And at the same time, then we're able to clone the genomic DNA, and to show that the various hormones, the glucagon, GLP-1, and GLP-2, are co-encoded in separate exons. There are 6 exons, of which 3 of the exons, exon 3, 4, and 5, encode glucagon, GLP-1, and GLP-2, respectively. Then the next step, after having gotten the roadmap, the structure of the hypothetical structure from the DNA, decoding to see if the peptides really how they're distributed in the intestine versus the islets. And so this is somewhat complicated result, but it was done by having developed antisera to the structures of the GLP-1 and GLP-2, which and then using the antisera to analyze the fractions from high pressure liquid chromatography separation, and could begin to put together a consistent theme where the primary peptides expressed in the intestine were the glucagon-like peptides, glucagon-like peptide 1 and glucagon-like peptide 2. In contrast, in the islets, the major hormone that was produced in the rat pancreas was glucagon, and the glucagon-like peptides were present, but at much lower concentrations than glucagon. And in addition, back in the intestine, the glucagon peptide is contained within a larger protein called glycantin, which had been identified in the intestine some years before. So what the peptide analyses by chromatography and immunoassay detection showed us was that there's a tissue-specific post-translational cleavage by endopeptidases called prohormone convertases. And so in the pancreas, the predominant peptide that's cleaved from the proglucagon is glucagon, and in the intestine, the primary peptide that was released are the glucagon-like peptides, particularly GLP-1. And it's done by two separate convertases, PC2 and PC1-3. And so we have the glucagon, which is a hormone, has anti-insulin, as we shown earlier. And in the intestine, the insulinotropic peptide, which is involved in regulation during the feeding stage, as opposed to the fasting stage where insulin, or glucagon is the primary peptide released. So a summary of these early studies on discovery and structure is that the glucagon and two distinct GLP-1s are synthesized in the form of a prohormone, which is called proglucagon or pre-proglucagon, with the signal peptide attached. And multiple peptides are cleaved from the proglucagon during biosynthesis via selective post-translational processing by the prohormone convertases. GLP-1s predominate in the intestine, glucagon predominates in the pancreas. But now there's a conundrum, and that is what the GLP-1 peptides are encoded in proglucagon are bioactive. As we showed in the earlier slide, there's a large array of different peptides produced in the both the pancreas and the intestine, but we needed to determine which ones were bioactive. And the structure of proglucagon was somewhat unusual in that it started with the two basic residues, arginine and lysine, or lysine and arginine, is a classic cleavage site for prohormones to produce peptides products. But so it started with the histidine, which is the hallmark starting sequence of glucagon, and glucagon-like peptides, and then went 6 amino acids, and this with a single arginine, then started at position 7 with another histidine, and went out this way. So the best guess at the beginning was that the cleavage would take here, and the GLP-1 sequence would be from 1 to the C-terminal, 36 or 37, because the C-terminal also had 2 different cleavage sites. One was at the double basic residue arginine, which produced a peptide of 37 amino acids, but with a G and another arginine allows for a second cleavage, but also an amidation of the C-terminal arginine, which may cause a peptide of 36. So there were 4 peptides possible, 1 to 36, 1 to 37, and 7 to 36, and 7 to 37. And all 4 peptides were cleaved from proglucagon by the prohormone convertase 1,3. So the studies needed to determine what the which was the active peptide. So the one study we did was carried out by with Dan Drucker, who was a postdoc in my lab at the time, and that is using cultured beta cells, a beta cell line, the Brin 104638 insulinoma cells, and then it was measured the responses of 7 to 37 and 1 to 37 on cyclic AMP formation, since we discovered, and others had discovered, that this receptor for GLP-1 was a G protein coupled receptor coupled to the GS protein that regulates via cyclic AMP signaling. So we measured cyclic AMP formation and found that the active peptide was 7 to 37. As you can see here, with a maximum stimulation of cyclic AMP at 5 nanomolar, and the 1 to 37 had no activity until you're out to 100 times higher concentration, which is probably artifactual conversion of some of the 1 to 37 to the 7 to 37. So then further can show that the 7 to 30, the 7 to 37 peptide, the active peptide, would also stimulate insulin production by showing an increase in the messenger RNA for proinsulin with the 7 to 36 peptide, the active peptide. And this is the control, and also stimulated insulin secretion in these cultured cells. Then became important to look at another system for measuring activity of the peptides. And here we had a nice collaboration with Gordon Weir at the Joslin Diabetes Center. And using the isolated rat perfused pancreas system. So it's an in vivo system, not cultured cells, but actually in a live pancreas. So we can show very nicely that the 7 to 37, the peptide but not the 1 to 37 peptide, stimulated insulin secretion in the perfused pancreas. You show here at 50 picomolar stimulation, which increases insulin secretion is increased when it's raised to 5 nanomolar, 100-fold higher concentration, and whereas the 1 to 37 has no effect on insulin secretion at either 5 nanomolar or 10,000 times higher than the 50 picomolar. At 500 nanomolar, there's no effect on insulin secretion. So the answer to the conundrum of what peptides are active was resolved in that the 36 and 37, but not the 36 and the 7 to 36, 37 are active, but not the 1 to 36 and 1 to 37. So we can show that the activity requires the cleavage of the proglucagon at the single basic residue, not the double basic residue. Likewise, it studies, and I won't show you, but there's the equivalent activity of the amidated versus the glycine extended forms at the C terminus. Now, I'm going to show now some 2 early studies on the three studies done on models of diabetes in rats and mice and in humans. The partial pancreatectomy rat model done by Susan Bonnerwehr at the Joslin Clinic, and the leptin receptor mutation, the DB mouse model of diabetes, which was done in collaboration with Josephine Egan, the National Institutes of Health, and the human subject volunteers. First, the partial pancreatectomy model could show using a hormone GLP-1 receptor agonist it attenuates diabetes in the partial pancreatectomy rats. And first, the plasma glucose shown after a 10-day administration of the exendin 4 analog of GLP-1 in the pancreatectomy mice who have diabetes, because you show the increase in glucose due to the removal of about 2/3 of the pancreas, so that they have markedly decreased beta cell mass, and therefore develop diabetes. And the administration of the exendin 4 GLP-1 agonist lowers the glucose. So it's a partial cure of the diabetes. And the sham surgery mice have no elevation of glucose. The administration of GLP-1 agonist exendin stimulates, increases the beta cell mass in both the sham and the partial pancreatectomy diabetic rats. So the effect on beta cell proliferation is shown here, and it's very small in the pancreatectomy mice, but surprisingly very high in the actually sham operated rats. So it shows the powerful effect of the agonist, the xenin agonist, on stimulating the increase in beta cells. Then I just wanted to end by thanking and acknowledging the contributions of many, both workers who put in much of the effort, and also the collaborators that we've had over the years have contributed to this work. And with a very special thanks to John Potts, who's been my mentor for many years, almost over 5 decades now, and also the support from the Howard Hughes Medical Institute, National Institutes of Health, the Diabetes Association, the Juvenile Diabetes Research Foundation, and Novo Nordisk. Thank you.